Okay, great. Thank you. So hi, everyone, and welcome to today's Friday noon talk. So we're delighted to have back with us today Richard Richardson from Canadian Nuclear Labs at Chalk River. So Richard started off his career in the UK with a BSc in Physics and Astronomy from the University of London, followed by an MSc in Medical Physics from the University of Aberdeen and a PhD in Physics from the University of Bristol. And Richard, I believe you worked in the UK for a little while before you came over here to Canada.
8 years in the hospital.
Okay.
Yeah. Medical physics, actually, was medical physicist.
Yeah. Good.
Good job.
And how long have you been in Canada? You've been in Chalk River for...
about 25 years, yeah.
25 years, okay. And Richard has given us many talks over many years, but we're always delighted to have him back. And today he's going to speak to us about the uniqueness of the multistage-senescence model of carcinogenesis and its potential application to radiation-induced cancers. So over to you, Richard.
Okay. Well, next slide, please. Okay, this talk I've split up into 6 parts. So there'll be a background on ionizing radiation and cancers of aging. 2-stage models of carcinogenesis come next. Multi-stage model and P53 mutations. And then go on to a more advanced model, the multistage-senescence model and the tumor senescence tumor suppressor factor B. Then more results on the model, and then talk about driver mutations and intrinsic cancer incidence. And lastly, the multi-stage senescence model applied to ionizing radiation. Next slide, please. So the first, as I said, backgrounds on ionizing radiation and cancer. Next. So this is a trip around generalized characteristics of cancer and aging. So can you hear me okay? Okay.
Yeah, so, we can hear you okay.
Okay. So there are 2 forms of spontaneous and cancers and ionizing radiation cancers. I'll call them developmental cancers and in fetus and children, and cancers of aging. Now you can see here, this is a rare cancer, but incidence per million there. Osteosarcoma rates, US data. You can see the first peak is it's in adolescence, in childhood, if you like, and the second peak to near the 70s, 80s is in old age. And this is a typical, so there's two basic types of cancer, but rarely does the childhood cancer dominate the cancer of aging. In fact, far the reverse if it happens. It does both. So to the right, the panel to the right has got colon cancer and lymphoma, non-Hodgkin's lymphoma, and then brain cancer. And you'll note there that the cancers peak around about 80 years of age and then decline to virtually 0. So that's the age-specific, I'll just move, the age-specific incidence rate per 100,000 population for 5-year age groups from US, this is from US SEER data. And you'll see a lot of graphs showing incidence rates against age in this work. So most cancer models only account for the rise in cancer incidence. And so that's one of the reasons going on to the multi-stage senescence model, in that it shows the cancer decline. Now, one of the things about aging is that there's a decline in reserve capacity as shown up. I just want you to see the trend there in nerve cord velocity, all sorts of things, renal and lung function. And later on, I will talk about functional mass loss in tissues and organs. And I guess what I'm trying to say is that this mass loss will be in parallel with what you're seeing here. Okay, next, please. So radiation-induced cancers, you've seen these graphs, I'm sure. The main cancer risk for radiation is from atomic bomb survivors. And on the top right, you see the cancers that ionizing, excess cancers, solid cancers, leukemias. They missed the first 5 years of data, so some of the early leukemia will not have been seen. And now there are deaths also from non-cancers. So the A-bomb survivor data shows more aging effects than earlier studies, the current studies, but there's still no prostate cancer. So, and also there are low levels of diabetes and dementia, and they're only seen at high dose. So there's differences between ionizing radiation and general aging. Children are 10 times more sensitive to solid cancers than adults, and that probably also applies to leukemias as well. But as I say, the data was not there in the A-bomb survivors. Next one. Now, these hallmarks here for aging and cancer, the aging ones are sort of square boxes, and the cancer ones in that rectangular. And the two green ones in the middle are the overlapping characteristics, the overlapping hallmarks. And you'll notice these papers are highly cited. And you'll notice that the two overlapping hallmarks are genomic instability, mutations, and the other one is inflammation. And these two hallmarks are highly associated. Genomic instability and inflammation are highly associated with ionizing radiation. You'll notice that aging has some counter hallmarks to those in cancer, and I'll just look at one. Top left, telomere attrition, top right, evading growth suppressors. Okay, and there's one HC4 enabling replicative immortality on the cancer side. So you have two that are common hallmarks and several each side of aging and cancer that counter each other. Okay, next. Now looking at this is a published article and in 2009, and in this case, I looked at the different aging effects of apoptosis, cancers, osteoporosis, and so on, and whether these were identified in A-bomb survivor, ABS, as shown here. And in most cases, you can see that they were. There are some exceptions. Autoimmune diseases, no. Chronic inflammation is an odd one because, yes, inflammation is known with ionizing radiation, but it's also ionizing radiation has been used to cure chronic inflammation. So, but the take-home message there is that ionizing radiation-induced aging happens mostly at high doses, if it happens. And A-bomb survivors lost 1.3 years per grade. Most of that by cancer, related to cancer. Now, amazingly, and this is always worth remembering, that the inflammatory agents, according to them, A-bomb survivors by 9 years per grade. Next slide. And of course, we all know that ionizing radiation is characterized by genomic instability. And I say that rather than mutation. So, and here is a graph that does show stable chromosome aberrations related to low dose from Chernobyl. And if you look at the figure, the top curve, number 2, is that for radiation, low radiation exposures, and the bottom one is for the control group. So there is some difference, but of nuclear DNA mutations in lymphocytes, but there is, then it's not significant in this case. But one of the things that is notable is that these mutations don't, they keep going up non-linearly with age. And that is very, very unlike the cancer incidence, which peaks and comes down in the older stalled. Next. So now the 2-stage models of carcinogenesis, these are the first ones. So Knutson's 2-hit hypothesis. This is probably the most well-known of all models, and rightly so too. In 1971, Alfred Knudson hypothesized 2 hits of both alleles of a key gene in retinal neuroectodermal cell led to childhood eye cancer called retinoblastoma. And then the tumor suppressor gene, RB1, was identified in relation to this cancer. Now, if you have one normal copy of RB1, you don't get a tumor formation, but 2, and you do. So in some cases, the first hit is in the germline cell. In some cases, it's in utero. And always the second hit, nearly always, the second hit is in infancy. Patients with this inherited one mutation of the 2 develop tumors earlier. That's a straight line in that figure on the left, whereas the curved line is for the 2 double hit. Okay, you can see there that little image on the right, upper right, shows the difference between hereditary and non-hereditary cause of induction of this particular cancer. All right, next, please. Now, a 2-stage model can be applied to childhood leukemia, specifically in this case, ALL, which is very rare, but is the most common childhood cancer. It's about a third of the childhood cancers. And particularly, it's a little bit higher in affluent countries, higher rates. So the Oxford survey of childhood cancers looked at X-rays dosed to the fetus in the 50s, 60s, and the risks. And a mean dose of 10 to 20 milligray to the fetus caused a 40% excess childhood leukemia incidence. Now, there's been a lot of reviews of this and so forth, and I won't go into them. But I'll just say that CT scans later in 2012, published in 2012, did show that there was a risk of leukemia and brain tumors, although, again, it's 1 in 10,000 heart CT scans. Just note the figure showing the incidence per 100,000 person years. In this case, ALL peaks around about 2 to 4 years old. Okay, next. So the 2-stage model of childhood leukemia is usually said to be first stage initiation, as shown on the diagram, and the second stage promotion. The first stage is genital or in utero, and the second stage is usually in infancy. And they have done tests to find that 1% of the first stage translocations or aneuploidies go on to the second stage and then cause an overt cancer. So promotion can be induced by infections. That's a well-known one for ALL. And I published a piece of work that's showing that thymus atrophy also promoted this ALL. Thymus enlarge with breastfeeding, and they atrophy also with infection. So that seemed to fit. So solid cancers in general have more stages, longer latent periods. They, instead of having 2 to 5 years as leukemias, their latent periods will be 10 plus. And the reason for this is that they have a need to vascularize and metastasize, as shown in the diagram on the right.
Yeah.
And so they have more stages than just, certainly more stages than just the simple childhood cancers. And from mice, ionizing radiation produces a first hit developmental tumors, and whereas the second hit is from ionizing radiation of tumors of aging. Okay, next, please. So now the multi-stage model and P53 mutations. Next. So a P53 protein that's coded by the, translated by the TP53 gene controls cell division and cell death. And it's called a tumor, it's a tumor suppressor, and it's called the guardian of the genome. And it's been written about probably almost certainly more than any other gene, 80,000 plus publications. And TP53, that's the gene, is the most lost gene in human cancers. 50 to 60% is currently the estimate for that, of all cancers, found in all cancers, the mutations. So ionizing radiation exposure can cause double-strand breaks. And then the P53 protein accumulates and can trigger cell cycle and senescence and apoptosis, and induce cancer if TP53 is mutated. So I did a study of TP53 mutation in big populations, world, Japan, so on, using IARC data, and also compared this with cancer databases. Again, GlobalCan, IARC databases. Okay, next, please. So this study in 2013 was based on the multi-stage model of carcinogenesis. This model by Nordling and Armitage and Doll in the 50s is shown in this slide. So the data on the right is Canadian data, actually, but it shows the age-specific rates of cancer incidence versus age along the x-axis. And you can see there that the ASR is modeled by A to the age to the power k-1. So that's the basic model called a multi-stage model. And the stages are k. K is the number of stages. And the exponent of that, when you do a log-log plot, the exponent is k-1. So in other words, if you do a log plot of, then you get the slope, and then from the slope, you can get the number of stages for any particular cancer type. Okay, next, please. So here's some data. I mean, I just picked some data. On the right is showing the cancer exponent against the TP53 mutation rates, and you can see that there's a strong relationship for worldwide cancers, and the even stronger one for Japanese solid tumors. So basically, and this is based on in the table, some of the data shown there, TP53 mutations For the cancers shown here, they vary between 6.1 and 31%. And the cancer exponents for incidence and mortality are shown there. And then the our incidence for each one, for each cancer type. And this is when plotted against age. So this cancer exponents there, so we get the cancer exponents, K-1, and that gives us these number of stages, and then they're plotted against the P53 mutation rate. So what that shows, it took me 8 years to figure this out. I didn't get it at first, I must admit. The more somatic TP53 mutations for cancer types you get with the higher number of stages. Okay, that's fair enough. But this is what I didn't get at first, that it likely indicates that mutations in this key gene can advance multiple cancer stages. So that's the key message here, that one mutation can advance several different cancer stages, especially such a gene as TP53 that has so many different capabilities that it can carry out in the cell. Okay, next. So here we go on to some recent work published in last year. So this work was published in Aging, a profound synchrony of age-specific incidence rates and tumor suppression for different cancer types as revealed by a multiple senescence model of carcinogenesis. And this work was with Catalina, Anghel, and Deng of CNL. Okay, next, please. So this describes the multistage-senescence model of carcinogenesis. So first, just to say that the multi-stage model without the senescence part by Armitage and Doll, and another model called the MVK model, which is you see Knudsen is it in there, that this two-stage model of clonal expansion, like the Armitage and Doll model, do not predict that cancer declines. So in other words, if you look at the figure on the right, you can see there the dashed lines and the undashed line just keep going up, and of course that's not correct. You need another model to bring the cancer rates down. And so what we use here in this work is the model of Pompei and Wilson, and they called it the beta model, and I call it the senescence part. So you can see the blue part of that equation there is the multi-stage model of Armitage and Doll, and then the red part, 1 minus BT, is the senescence model. And T is age, K is number of stages, and B, I call here the senescence tumor suppressor term. All right, so that's B, and that brings those cancer rates down to 0 around about 100. Next, please. So there's a big job fitting the data from SEER, US SEER incident data available to everyone, 2000 to 2003, and data for 2010 to 2013. And the cancer types are given in the form of TCGA types, as defined in that atlas noted there. The male model fits were to 24 non-reproductive non-sex cancers and 2 reproductive, which I call sex cancers, by methods of Harding et al. Published in 2008. Female model fits were 24 non-sex cancers and 5 sex cancers. And we omitted rare cancers or bimodal Hodgkin's lymphoma, which, it's got a childhood part and a aging part. So next, please. Here's some data. It's just to give you an idea of what data was collected. So the first column, sex, male and female, male and female, as you can see. The cancer type, bladder. I'll just say what they are basically. Bladder, colorectal, lung, skin, prostate, and breast. They have a 4 BLCA as the bladder cancer that we're looking at here. And case stages, they vary between 3.7 for breast and 8.5 for bladder here. Senescence factor, look how close they are, especially the non-reproductive ones, non-sex, they're all around 0.01. And the age at peak given, and the next column, next green column, stage transition rate, then the cumulative probability. And if you go down to the bottom one, breast cancer makes up 19% of all the cancers. That's what it means. And the last is to do with, well, we'll come on to that later. Okay, last column. Next. So profound, what we can say is there's profound consistency in the senescence tumor suppressor, B. And you can see it in the equation there. The mean value for male cancers is astonishingly tight, 0.0098, plus or minus 0.0008, and is insignificantly different from females, which is a 2% difference. Now, the only one that really is different is when you have look at the 5 female sex cancers, the senescence tumor suppressor term is significantly higher. And when you look at all cancer terms, the senescence term goes to 0 when T is 98 years old. So if you get to 98 and a female, you do well. And by the way, that's why the very old should get their pneumonia shots, because this cancer is not what the oldest old, as they're called, died from. Okay, and 94 years for males. Next, senescence tumor suppressor and mass loss of tissues and organs. Now, what is causing this red portion of the multi-stage senescence model? And because this is 1 minus BT is the senescence part. Well, that B factor, B per year, decline in the cancer rate there, times the age, is similar in value to the serum insulin-like growth factor. That is a very prominent growth factor, IGF-1, decline per year in adults. Now, you may well know about the IGF-1 deficiency, which is called the Laron's syndrome. And there are individuals living in the Amazon, I believe, that have short stature, but they also have developed no cancer, none at all. And so this growth factor may be important. Well, it will also affect tissue mass. So the study of functional mass loss, which I did published in 2014, accounts for increases in fat, fiber, and senescent cell content. And you can see there, the graph on the right, upper right, that mass change is highly correlated with cell turnover. And so, and also the other thing that was found was the onset in major human organs is between 21 and 35 years of age. And of course, that shouldn't surprise you when you also look at the physiology, as I show you in the third slide earlier. So what you can say is that 70% functional mass loss variability in human organs is related to the cell turnover time. So this sort of makes sense in an evolutionary way, in that it's deleterious, and rapid cell loss, cell division will cause shorten the telomeres, potential cancer stem cells. You get to the Hayflick limit, it'll trigger cell death and senescence and leads to function. But the downside is it leads to functional mass loss. The benefit when these tumor suppressor genes are not mutated is that it suppresses cancer at the expense of sarcopenia, osteoporosis, frailty, all commonly associated with aging. Okay, next. More results, including driver mutations and intrinsic cancer incidence. Next. So the model, the multi-state, the fits that we talked about earlier, here you're looking at the age of peak incidence for model fitted and to SEER data for males and females. And the age of peak incidence was from a derivative of the equation seen there, ASR. I'm not showing that. The relationship, non-significant relationship you see in figures A and B is highly significant. So there is a strong relationship between the age of peak incidence and the number of stages. So what that says is that the incidence of simpler cancers with fewer stages peaks earlier in life. Because we know that, we already know that childhood cancers have fewer stages and they peak very early in life, but also other cancers such as thyroid cancer and glioma also peak earlier than some of the cancers like colon or bladder cancer. Okay, next. And another thing from this model, now a little bit more maths here. The multi-stage theory can be broken down a little bit. The A in the equation shown earlier can be broken down into probability. And from that, you can estimate the stage transition rate. Now, as K assessed by the methods used here is not an integer, it's not a whole number. You have to account for this by using a gamma function, which is gives you a non-integral okay, I'll continue. What we have here is that this shows that what we have is a linear, very, very strong relationship between mean stage transition rate and the number of stages. And it's shown there first for males, A, and females, B. And so what we can take away from this is that complex cancers with high number of stages have faster stage transitions. So this U value, which is stage transition rate, for male cancers on average is about 1/3 greater than that for females. And that was derived from this study. So that helps explain why men have 73% greater lifetime risk than females who are developing a non-sex cancer. Okay, next. Now, looking at this equation again, the multistage-senescence model, what you can do is from the previous figures, you can derive a two-variable non-sex cancer model. And I'll just say that, and the constants are given here, B, C, and D. And from those, you can produce a figure such as on the right-hand side. And this is illustrative in nature, in that what it shows you is that for a small number of stages, thyroid cancer, say, with K 2.7, the peak incidence rate is lower and it occurs at a younger age than complex cancers such as bladder cancer, where the K is 8.8. No other model of carcinogenesis predicts stage-related trends or related relative magnitude of incidence rates for different cancer types. Okay, next. So I'm not going to go into this in great detail, but the two-variable model gives us the intrinsic cancer risk. And so how do we know that? Well, intrinsic cancer risk is defined as the lowest incidence level without exogenous non-intrinsic factors, such as there's no smoking, no obesity, no ionizing radiation. And the intrinsic cancer risks have been estimated by Wu et al. In Nature in 2016. So what we do is compare that cancer risk with extrinsic cancer risk, which is from our SEER data. That's the data from the US, and it has some of these factors in. There are smokers, there are and other lifestyle factors that induce cancer, alcohol, obesity, and so on. So what we found was that the two-variable model has a strong association with intrinsic cancer rates, details in the paper. Our highest extrinsic to intrinsic risk ratio was 40 for prostate. Now, that seems outrageous, and 30 for breast cancer. But and that's given in the last column in red on the table that you saw earlier. If you look at the lower one for breast, lowest row, and the last column, you'll see that the ratio is 30 for breast cancer. But looking at prostate cancer, this graph published in 2016, this figure on the right-hand side, bottom right, shows the incidence and mortality rates for prostate cancer. Look at the difference between developed countries such as Australia and New Zealand on the left, with the highest rates, and those on the right in Asia. And basically, the highest risk is with highly developed countries. And I won't go into, I'm not an expert in prostate cancer, but reading around it, it is very much related to the Western diet, and there's other factors too. Okay, next. This is a bit of an aside, but it still uses the data from the paper, in that here we plot in the diagram on the right, in the figure on the right, the number of driver mutations against the number of stages. And just before we go to explain it a bit further, cancers have hundreds to thousands of passenger mutations and very few driver genes. So the passenger mutations are due to genomic instability, and the driver genes are what drives the cancer forward. And the driver genes are usually mutated oncogenes and tumor suppressors, such as P53, or as we've shown earlier. The figure on the right shows complex cancers with more case stages have a higher number of driver mutations. If you take the data for 20 TGCA cancers, and this is used in the data of Lorenzo et al. In 2018, you could show that driver genes contribute 1.7 of the 6.2 stages. So basically, rounding up this, the average cancer has 2 driver genes mutated, but 6 stages in all. And so 1/3 of the cancer stages will be driver genes. But this does not count for multiple stages via TP53 or KRAS tumor suppressor genes. what are these non-driver gene stages likely to be? Well, epigenetics, inflammation, metastases, there are other factors too that are well shown to advance cancer. Next, please. Okay, so finally, the final part is multistage-senescence model related to ionizing, applied to ionizing radiation. Next. Okay, so here we're looking at the tissue weighting factors according to ICRP 103. I've omitted gonads, and because it occurs not in the present lifetime, and the 4 tissues of low weighting factor 0.1, and also the remainder tissues that together make up 0.12. So in the first column, the tissues, red marrow, so induces leukemia, is where leukemia will be induced. Colon, lung, stomach, breast, and then some at the lower weighting factor of 0.04, that's the second row, bladder, esophagus, liver, and thyroid. So these show the most prevalent cancer after radiation exposures. Now this, I've given in the third column GCA cancer types according to the SEER data that we employed. And then the case stages are shown for that data. So they vary. You can see their thyroid 2.7 there, and up to 8.5 for bladder. And in summary, ionizing radiation, the mean weighted number of case stages for 76% of the weighted cancers here is 5.3 plus minus 4.9. And for the SEER most common cancers, I take 77% of them, of cancer incidence accumulated over a lifespan. The mean weighted number of case stages is 6.4 plus minus 3.9. And the result is that the number of stages for ionizing radiation cancers is not significantly different from common spontaneous cancers. That's not always the case. On some work that we're looking at, that I'm looking at, is that smoking doesn't act in a similar manner. I can say that. But the great study by Heidenreich et al. In 2002, they applied all sorts of models, but I'm giving the result here for the Armitage-Doll multistage model, 6 parameters, note, not 2, to incidence of solid tumors to A-bomb survivors, and estimated 5 to 6 stages for females cancers and 11 to 12 stages for males. Now, we've shown that from the data above that it's very, very unlikely that radiation causes a doubling of the number of stages. You would have to go on to different cancer types entirely. And somebody, I didn't get the references yet, but somebody told me that radiation doesn't have this kind of characteristic as producing entirely different cancer types. That's not true of other carcinogenic agents, but is for radiation. So from this, we can say that it's likely that ionizing radiation increases the stage transition rate, not the number of stages. Okay, next. And last, in summary, So in summary, the senescence tumor suppression is remarkably consistent across cancer types. So it probably has an evolutionary nature. Cancers with fewer stages have longer stage transitions and peak earlier. Driver gene mutations contribute about 1/3 of the stages of all different cancer tumor types investigated. A 2-variable model, age and stage only, is strongly associated with intrinsic cancer risk. And uniquely, it gives the relative incidence of different cancer types. The 2-hit initiation promotion models are inadequate for most cancers of aging, spontaneous, or radiation-induced. Radiation-induced cancers, usually the aging ones, are likely due to the increased stage transition rates. And this is also true of the high spontaneous risk of cancer in males. Okay, thank you.